Well, thank you. It's a great honor to be invited to speak at Yale with this tremendous legacy, and particularly to give the Iris Streiss lecture, with obviously the tremendous legacy of Dr. Strauss here. I actually changed the title slightly. Sorry about that. I mentioned to sort of hone down on what kind of the major focus of the talk is, but certainly it'll be involved in precision prevention and other aspects. I'll start out with work in the head and neck area, and sort of the first part of the talk, I'll go through a lot of sort of my career in head and neck in the area of prevention, particularly chemo prevention, and why we're moving very heavily towards a different kind of approach, immune-based approach. But this is the model system that we studied quite a bit when I was at MD Anderson, and that's oral premalignant lesion progression model. So clinically, this is on the buccal mucosa. You can see a whitish lesion progress to cancer here. Histologically, we knew that the evolution of hyperplasia, increasing grades of dysplasia to cancer. And at a molecular level, we did, certainly the techniques and technology that exists now didn't exist then. And so we spent a lot of time as a group studying various aspects of the evolution and molecular progression model from oral premalignancy to cancer. This summarizes a series of 3 randomized controlled trials, abbreviated RCTs, that we did with the drug 13-cis-retinoic acid, also called isotretinoin. And you can see the trials were what we thought a logical series, from a high dose of the drug for short term versus placebo to sort of a high dose induction followed by longer term maintenance, and then finally the most recent trial, which was published a few years ago now, that actually looked at a longer term low dose intervention with different retinoids, and I guess the point I want to make here is that this was a fairly disappointing result. Because it's a complicated story. It was a 2-arm trial. The third arm evolved in the middle of this trial, which was NCI funded through a PO1. The work had just come out indicating that beta-carotene was a co-carcinogen with cigarette smoke, increased the risk of lung cancer in smokers. So the NCI made every study involving beta-carotene close and look at very careful safety issues. Anyway. So when we started, it ended up pulling beta-carotene out of it. But the point really here is that this is actually looking at cancer development, not reversal of a premalignant lesion. So it's not shrinkage or histologic improvement of a premalignant lesion like leukoplakia in the mouth. It's actually looking at the development of cancer, and the 13-cis arm, which is the key drug that we were interested in, you can see here, was not different than either of the 2 therapies, retinol palmitate or the combination of beta-carotene, retinol palmitate. Again, we had to get rid of beta-carotene because of the NCI's concern about the toxicity. So this really made us, this is a couple decades of work here, and made us sort of pull back and rethink things in the head and neck. But it sort of led to a large part of my career, which is sort of characterized by negative randomized trials. So we came up with, someone came up with a new term of important negative trials. So you'll see why I got to where I was. So I did have a couple of lung trials here, because I know it's a big lung group with Roy and Rogerio and others. But so I pulled this one. I almost forgot about it. But this was a randomized trial of the same drug in stage 1 non-small cell lung cancer. It was the largest randomized trial in this setting. I don't know if it still is, but at the time it was the largest randomized trial in stage 1 disease, and the primary endpoint was the development of second primary tumors, shown here. And you can see there's no difference in second primary tumor rate, recurrence, or death in the 13-cis arm, which is in red, versus placebo. A subset analysis, it was a pre-specified subset analysis, is shown here. I just took this out because this generated a lot of discussion. This is actually the mortality, and it turns out in the subgroup of people who were current smokers and took the drug, the overall survival was significantly worse in the treatment group. The placebo, this held up on multivariate analysis. And based on this subgroup analysis, which we didn't make a particularly big deal about, in there led David Johnson, who some of you know, who I guess is head of medicine now at UT Southwestern, in a very public forum at ASCO, to say that Scott's been able to do something that no one else has done in the history of lung cancer, and that is to make lung cancer worse. But in any case, we don't recommend, so I got out of this thing. There's a couple other trials here, but lung is a tough one. And then, so most of my areas in lung and head and neck, and I chaired that department at MD Anderson with Roy and others, but I was for a while chair of the Cancer Control Research Group in the Southwest Oncology Group, and they have a very big prostate group. So even though clinically as a medical oncologist I just see head, neck, and lung patients, I was involved in a number of large prostate trials. This is one of them called SELECT. It's crazy. I don't know at what point in oncology we became fixated on acronyms. Everything has got to have an acronym for a trial or it doesn't work. And I remember sitting in a room in Seattle for hours where we each came up with different acronyms, and mine always turned out to be a little X-rated and never got picked. But in any case, it's selenium and vitamin E cancer prevention trial. But this also is, this randomized trial involved over 35,000 men. I think it is the largest randomized trial, prevention trial ever. And we can go in why we did this. People criticize this quite a bit. People come and sort of accost me at ASCO saying, "Why are you doing this? I can't get an R01 because of all the money you're putting into this large trial." And not that it was a correlation, but it was important because at the time we started this trial, people just believed that vitamin E and selenium prevented prostate cancer. In fact, there were taglines on that runner, whatever they call it, on CNN: "Take vitamin E and selenium, prevent prostate cancer." So there was this feeling. We can go through why that is, but to do it, and we felt it was important to study this issue. Anyway, a large trial, and you can see it had 4 arms: factorial design, placebo, vitamin E, selenium, the combination. And this is probability of prostate cancer, which was the primary endpoint. And you can see here this top dotted line is selenium, is vitamin E, excuse me. So you can see here that vitamin E is separating from the group. It was not significant, but there was an increase in prostate cancer development in the people who got vitamin E. So to tell you how important this negative trial was is that we just literally extended follow-up for 2 years, and JAMA published another big paper on it just because this trend now was significant. It was here, and now it actually reached a significant p-value. So again, redefining important negative trials. And again, this is what, I don't want to be facetious about it. This is what really prevention was largely about at the time. A lot of trials, and I was a big part of this, so I'm not blaming anyone, but a lot of it was sort of the Wilford Brimley idea. If you remember when he was on a horse and would be advertising for grape nuts. They still make that cereal, but in any case, he said, eat grape nuts, it's the right thing to do. This was kind of the approach. Vitamin E, selenium, it's just the right thing to do. How can it be bad? Well, we find that actually these things not only may not work, but can be harmful. So we moved into, really after that last study in head and neck that I mentioned earlier, into looking at targeting genetic drivers and thinking about prevention differently. Instead of just characterizing premalignant lesions based on how they looked or the histology, we wanted to dig a little deeper. This is a slide that's borrowed from Linda Chen, with the idea that genetic drivers, we know, obviously exist in advanced disease. Lung cancer is a great example of that. And they can be found in early disease, and in some cases even cancer, even pre-cancer. So what about if we try targeting some of these genetic drivers, or at least identifying them for the risk? And This work, this shows you the evolution of loss of heterozygosity profiles and the evolution. And actually, the initial paper on this was from Li Mao when he was in our group at MD Anderson 20 years ago now, looking at those same kind of premalignant leukoplakia lesions in the mouth and characterizing them not by histology, but by whether or not they had loss of heterozygosity at specific sites on chromosome 3p or 9p, sites of presumed tumor suppressor genes at the time. And you can see here that if you had loss of heterozygosity at one or both of these sites, your cancer development rate was much higher than if you had retention. This has been confirmed. This is a group in Vancouver, and the most recent paper is published here with this logistic regression model showing the different patterns of loss at 9p and 17p, which is p53. And it was a very nice study that had a prospective cohort, so that literally in that risk of those patients with those lesions, you could characterize people with very low risk. This is in years at the bottom on the x-axis. Intermediate, low, intermediate, and very high risk, and so it's an opportunity to focus on these really high-risk individuals. It's a big issue with prevention, right, because they don't have cancer, and identifying high-risk groups has been a big challenge. So we took this data and developed the EPOC trial, or Latin Prevention of Oral Cancer trial, funded through an NCI P01, and published a few months ago. And this was a very complicated trial to do because instead of taking all comers the way it was before, you have a lesion, you're biopsied, you're on trial. Now we actually registered all comers, but they had to undergo real-time LOH profiling with turnaround within 7 working days. And then based on that, if they were LOH positive, they were eligible to be randomized, and ultimately it actually turned out exactly as designed. It was supposed to be 150 randomized patients, 75 in each arm. Eligible, but it was a very complicated. A lot of patients needed to be screened and registered to get this trial going. It's interesting. It came out with a editorial from Jenny Grandis with basically saying this was sort of the beginning of an era of molecular selection that had not been done in the prevention setting before. Obviously, not the same as targeting genetic drivers, because this was a risk marker. It's obviously difficult to target loss of heterozygosity, but at least selecting patients based on molecular features. And the primary endpoints shown here, there was no effect of the drug shown in black. There was this interesting pattern that suggested some benefit early on, and I didn't show you this, but this was only a 1-year treatment with erlotinib, and it was full dose erlotinib, the same dose we use for lung cancer therapy. But when the drug was stopped, the effect wore off completely. The only significant finding is shown here, and that is the development of a rash within a month. And a lot of us say, well, yeah, I know that. Lung cancer, it's been shown, and head and neck cancer and so on. It hasn't been shown in a prevention setting, and to my knowledge, we don't really know the basis of that. But given all this interest in immune oncology, it's possible that this is reflecting some sort of immune response. But we don't know that. But in any case, much better cancer-free survival if they had a grade 2 or greater rash than if they didn't. We were able to refine certain and prospectively validate certain risk profiles, LOH, EGFR copy number, and showed that individuals that had both loss of heterozygosity at certain profiles and had EGFR copy number gain had the worst prognosis. And we did a number of different subset analyses. Unfortunately, I mean, we were, obviously we were interested to see if the group that was EGFR FISH positive had the target of the drug might have done better, but in fact there's no evidence of that. And as we know in lung cancer and others, the FISH positive story just didn't pan out as a predictor. I just want to point out that it's interesting that in a early analysis of the TCGA for head and neck cancer, the importance of p53 mutation and loss of 3p came out of a very comprehensive computational biology analysis, Trey Eidecker's group at our place, it's interesting. So again, using these particular sites of 17p and 3p LOH. Seem to be important prognostic factors in cancer as well. And this is interesting in that if you look at, again, loss of heterozygosity at 3P or P53 mutation, you see that retention at both sites is associated with the best survival. And if you had a P53 mutation, you were here. If you had P53 mutation, you were here with 3P retention. If you had 3P loss, but wild type P53, you were here. But what really separated out was the group that had both loss of heterozygosity at 3P and P53 mutation. What's pretty fascinating, I think what actually got this paper in to Nature Genetics is a finding that head and neck people will know well. So we actually studied the subset, this is within the TCGA, of HPV positive disease. And those of you, Barbara and stuff, know that there aren't a lot of HPV positive patients in this particular TCGA data set. But here you see that here's HPV positive disease in terms of survival, and this is HPV positive disease with 3P loss of heterozygosity. And obviously this makes sense because P53 doesn't really play a role here because HPV inactivates P53. So it certainly was consistent with this finding above. And I just want to put this up because, you know, these days, I mean, obviously we're all investing a lot in computational biology because, you know, the data is out there. And that paper that I showed you, the Nature Genetics paper that Trey Attiker from our place did was actually published. It was the first paper published from the TCGA. It came out before the primary paper, which is shown here, again, with the ability to mine the public data sets. And then sort of finally on this topic, you know, looking at other potential targetable drivers in head and neck or premalignancy. And of course, the challenge in head and neck, which is very different, for instance, than lung adenocarcinoma, particularly never smokers, is that most of the drivers are tumor suppressor genes, so inactivated. So it's really, there aren't any really great targetable genetic drivers. Notch1 and PI3K-CA are ones that people are interested in. Notch1 is tricky. It can be either a tumor suppressor or oncogene in different cases. But you can see the different sets in the TCGA, and we actually looked at this in a subset of patients in EPOC and found this kind of pattern that, again, the PIK3CA looked like an interesting target. If we had good agents to target this, that might be good for head and neck and potentially for premalignancy. And then this is, you know, it's even hard to show this because it's pretty anecdotal, but with all the interest in immune prevention, our pathologists looked at some of these samples, these premalignant samples, looking at with immune profiling. And it's interesting here that, this is a dysplastic premalignant lesion from EPOC. And you see here, this one had EGFR gene amplification. And there seemed to be, again, I hate to use this loosely because there are only a couple that were truly amplified. But in any case, here is one case where you saw a dense lymphocytic infiltrate associated with amplification of EGFR, which, you know, has potential interest in immunoprevention. Okay, Roy, I said I would put some of the slides here that I pulled out that you led. This is one of those in a review article looking at molecular origins of lung cancer in either smokers or adenocarcinoma non-smokers, and a series. I'm sure you've heard a lot about the different battle trials and so on in advanced disease. What I'm just showing a slide is on early disease, premalignancy, and it's interesting that, you know, for a long time we thought that the whole effect of in smokers or just in general was that there was a field effect, and that it was field carcinogenesis, and that you would think, if that's true, let's just look at a smoker, that you would think that you would find premalignant evidence of premalignant disease at a molecular or cellular level throughout the lung and so on. But it turns out that, at least in this case, it's more of a clonal patch as opposed to a diffuse infiltrate. And so this is work that I'll show you. But in non-smokers that have adenocarcinoma, you can find evidence of a clonal patch of premalignant disease. And in this case, I'll show you, is with EGFR. And as you probably know, the EGFR mutation, the activating mutation, is one of the earliest changes. It precedes FISH changes. It's one of the earliest changes you can see. It occurs in premalignancy. And this is data that's kind of older now, but still really important, I think, from Ignacio Westuba's group, where he actually looked at lung cancer resection specimens and found that in patients whose tumors had known activating EGFR mutations, that when he looked at a histologically normal epithelium within the tumor, 43% had mutations. And if you look at the adjacent area around the tumor that's histologically normal, you find that there still are mutations, but they're less. And it's very infrequent elsewhere, and you don't see EGFR mutations in patients who don't have EGFR mutant tumors. So again, EGFR mutation is early. It occurs in histologically normal disease. You know, it's and so I think it potentially could be something that could be targeted. There are many challenges identifying who has these lesions just based on risk modeling. So now moving into immuno-oncology. Obviously there's been tremendous interest in this in therapy, particularly with the immune checkpoint inhibitors and in prevention with vaccines. And I feel, after spending a long time in the field of chemo prevention, that there have been advances, but I would say most of them have been incremental at best. And the idea of chemo prevention, whether it's aspirin, tamoxifen, what have you, drugs that work, but the idea of having to take a drug every day for years is a real challenge and produces a lot of side effects and other problems. So I think if we can bring what we've learned about therapy and how immunotherapy works, and in prevention, the equivalent of that would be vaccines. I think giving immune checkpoint inhibitors or other kinds of CAR T therapy are obviously not going to work in someone who doesn't have cancer. So the equivalent of immunoprevention essentially is vaccines. And we know that if we know enough about what the driver is of premalignant lesion, what's causing it, they can be very effective. So HPV vaccine is the model. We know there's a single driver, a cause of cervical cancer and other cancers. There's known risk factors, so you can give these truly prophylactically. You know, not a lot of times we think of prevention, someone has a premalignant condition. Here we're talking about before even exposure. The response required are neutralizing antibodies, and there are no immunosuppressive signals or cells in the microenvironment. And this is a dramatic finding. You can see here, we're not just talking about causing 99.7% of cervical cancers, but a large percentage of oropharyngeal cancers, anal, and many other cancers. In this nice review by Howard Bailey and an ASCO statement recently on this. Okay, so how do we take that information? How do we take the fact that immune interventions have produced important advances in therapy, vaccines and immune intervention and prevention is very effective when we understand it well. How can we take the cancer vaccines in this setting? And cancer vaccines haven't worked well in advanced disease. So a lot of the investigators in the cancer vaccine world, including Medav and others, are really moving more towards prevention. But there are a lot of challenges, and here are just some of them. So number one, the central drivers are generally unknown. There's not a single cause. And in general, you can't vaccinate until the premalignancy is identified, so you have this immunosuppressive environment, and it requires a T-cell immune response. So the challenges to moving vaccines in a prevention setting in a non-viral setting are that we have a poor understanding of the biology of premalignancy, the neoplastic cells themselves, and we have a poor understanding of the immunosuppressive microenvironment and their interactions. And so it's my feeling that we need to put a lot of resources into studying the biology of premalignancy. No one would talk about any cancer here just in terms of morphology as the only thing, I don't think, these days, right? We're all talking about signaling pathways, molecular subclassifications, and we're treating on the basis of this. Yet in premalignancy, we're still, it's like the dark ages, we're still using morphology. That's how we're characterizing it. So and that kind of approach is not going to help us develop vaccines to solve these problems. So a lot of interest by people in the field to do a PCGA, pre-cancer TCGA-like effort, like was done with cancer, but including the microenvironment. So this gets us, if you think from an immunologic point of view, to think about the biology of premalignancy in two ways. One is the classic molecular progression model. The neoplastic acquisition of mutations in oncogenes and tumor suppressor genes to develop cancer. but also to look at the antigenic repertoire within a premalignant lesion that may not include genetic drivers. It could include glycosylation or any targets that could be vaccine targets. And these can and so a lot of interest in the germline here. I'll talk a little bit about hypermutations and the MSI story, somatic changes, genomic, epigenomic, viral, and then omics-defined inflammation. And so this is just in this cartoon. These are antigen-primed T cells with the vaccine. And here you can see part of the immunosuppressive microenvironment, the regulatory cells, the M2 polarity, and these things are microbiota. And then here you see the viruses and so on. But in any case, this doesn't necessarily reflect drivers of the cancer, but they are potential antigen targets. So starting with the molecular progression model, and this is, to me, just amazing when I first heard this, actually from Bill Haidt when we were at some sort of meeting. And you know, the whole idea of the molecular progression model in cancer evolved, at least from my perspective, from the seminal work of Bert Vogelstein almost 3 decades ago. And you know, I just pulled this figure right out of the paper. I mean, you can't even get a good copy of it. It's just this is an old finding, and it really revolutionized the way we think of cancer. Yet, despite this, there have been a total of 25 sporadic polyps, sporadic adenomas, that have had next-gen sequencing. 25. And you can just imagine what it is in other premalignancies. So I think part of the issue is that, you know, there's thousands and thousands of patients in each cancer type that have had fairly extensive whole exome or other types of next-gen sequencing, yet very few premalignant lesions have been studied this way. And for several reasons. I mean, one, the technology wasn't there. This is very, very difficult to do. It's very labor-intensive to microdissect premalignant cells and apply these technologies on very low tumor volumes of cells. But the technology exists to do this now, and it's starting to be done. And this is sort of a recent example of just looking at. So here, this shows you the genetic drivers that Bert Vogelstein reported in 1990. This is what it looks like now. And this is just the genetic drivers within this early to intermediate adenoma phase. So you went from 1 to hundreds. So right away you're sort of opening up this black box, and now we can start to understand what makes premalignant lesions tick to that level. But it's really been a long haul to get here. This is just some examples of applying next-gen sequencing, single-cell analysis to premalignancy. Just a couple recent reports showing this process and identifying a new premalignant lesion in the evolution of melanoma. I'm not going to comment about this, but just it's interesting that maybe there are questions that these driver mutations not only can occur early, but they can occur in completely benign conditions. So if you talk to anyone who does melanoma, they all know this, that BRAF mutations are frequent in benign nevi. So that makes you think about, that introduces a couple issues, and we wrote a report on that in JNCI recently. This is in esophagus, this is Barrett's esophagus, and again published recently. There were 2 papers, I'm just focusing on one, showing that here where you seem to have a more linear progression model, in Barrett's esophagus you have different patterns. You have clonally related patterns, that is comparing the cancer to the precancer, but you also have completely unrelated. So in other words, the genomics using whole exome sequencing of the premalignant Barrett's esophagus is completely different than the adenocarcinoma of the esophagus. So again, illustrates some of the complexity at different sites. And then mutation order is also important. This work from Nelly Polyak with single cell techniques. This is just using breast cancer and BRCA1 mutation carriers. And the point here is that, the order of the mutations is key, that patients who end up, their first mutation is P53 mutation, and then have BRCA1 somatic loss, have a less aggressive tumor. Those that have a P10 mutation first, actually, with a similar mutation pattern, actually have the more aggressive basal tumors. So again, it just tells you how complicated this is. And here we are using morphology to characterize premalignant lesions, and here we can't even say that the different mutations that exist are important. It's not only that, but the order in which they're acquired could be very important. You know, again, this is an immuno-oncology, but it relates to the idea of the biology of premalignancy. and which was sort of the basis of my initial title. But since I'm focused mainly on the immune, but I wanted to just point out, so early detection is also being advanced quite a bit now by genomics. These are the first two, these are the only two validated genomic markers for early detection. And this is in colon, and this is from Avi Spira in Boston, in lung. These are FDA approved for early detection. There's some really out-of-the-box approaches to early detection. This is work that was led by Dennis Carson, who is a former director at Moore's Cancer Center, looking at developing new bioinformatic algorithms. Essentially, essentially the approach was to see if one can detect ovarian cancer in a pap smear. So this is kind of out-of-the-box thinking, which is kind of cool, I think. So obviously the pap smear is very effective for cervix cancer. But now that the technology is so sensitive and specific, they identified 6 specific mRNA isoforms, and they were looking to see, and we have a clinical trial now to do this, to see if you can detect these isoforms in a pap smear, because literally one copy through the fallopian tube may be enough to detect. And this would be a tremendous advance and a totally different way of looking at early detection. Then you have liquid biopsies. This is recent work from David Sudransky at Hopkins. And this is a huge challenge for using liquid biopsies for early detection. A huge challenge that people don't talk about. They talk about the technology, right? It's sort of why some people don't like the moonshot analogy. That, this is not a technology. I mean, I think the technology will get there. Again, when I started out in college, there weren't computers. So I think that, I think that technology will get there. But the problem is the biology. And here, we're starting to see that people with pre-malignant lesions, you can detect driver mutations in the blood in those patients. So in this case, it was a BRAF mutation detected in, well, not necessarily, but one of them was a BRAF mutation detected from an atypical adenomatous hyperplastic lesion of the lung. So this is why, big programs like this, get me sort of nervous in these, private, the public marketing. But some of you know about this Illumina spin-off company called Grail. Got a lot of press, as you can see. And the idea here is that they'll draw one drop of blood and be able to tell if you have cancer and where it is. And a lot of people who know the technology better than I do I'm not sure we'll get to that degree of sensitivity in a healthy person, purely healthy person. But even if you could, and I believe I believe technology can do anything eventually. How do you know where it came from? If you find a BRAF mutation, how do you know where it came from? It could be a premalignant lesion in the lung that you'll never see, and talk about challenges and so on. So this is, why I think if we understand the biology of premalignancy, it'll be easier to interpret some of these kinds of results. Applying next-gen sequencing is defining new premalignancies. This is just about a year and a half old. A new premalignant state called clonal hematopoiesis. And this was identified by repurposing next-gen sequencing that was out there. So, for instance, in this paper, they looked at TCG cohorts from 11 solid tumor sites, and they used the control blood. So this is the blood that, when you do somatic sequencing, you use blood just to subtract out the germline. They used the blood in a different way, which we can talk about, and they identified an age-related increase in certain mutations that are associated with myeloid leukemias. And then there were back-to-back papers last December 2014, I guess, and which were very confirmatory with this and with themselves that actually in the blood of healthy people, these are healthy people, there's an age-related increase in the mutations that are frequently seen in myeloid leukemia. So there's a lot of interest in the heme literature and the heme world on this fairly new premalignant state. And again, it's just interesting work because I think this is one that came from Roger Ebert's lab, and his lab people sort of laugh about it because it was the cheapest New England Journal paper they ever had because it didn't require any additional sequencing. You just literally used what was there to look at it a different way. And then so this field has evolved quite a bit. Raphael Behart at our place is leading this. He trained with Ebert, but you can have this initial the clonal hematopoiesis, idiopathic, I showed here. Now you can subset that into clonal hematopoiesis, either at diagnosis or in patients you know you progress, and then see what happens in myelodysplastic syndrome, and you can see that the number of mutations increases and so on. But again, there's a lot of interest in this new premalignant disease and following this and how to approach it potentially with immune approaches. And then somatic germline interactions. If you remember that cartoon where I had germline, somatic sort of as layered on. A lot of interest now in how the germline can influence somatic changes. This is in myelodysplastic syndrome. Just one good example. Germline got a 2 mutation affects what happens somatically. So of course the germline mutations in all cells, but it turns out there's some sort of interaction with ASXL1 somatically in the cells. And then eventually monosomy 7. So this germline mutation somehow predisposes to AXL1 mutations, and it's a very different pattern that you see there. So again, we have to look at the germline more than just the way we do now, but how it affects somatic changes. And this work from Hannah Carter, Trey Eidecker, looking at trying to do this in a very systematic way, and with bioinformatic approaches, looking at this widespread influence of the germline on somatic events. And it's interesting what can be uncovered when it's done less than an anecdotal way. For instance, it's interesting that FAT1, this is the first time that FAT1 mutation has been associated with higher mutation rates in TCGA. So I think a lot to be learned here that could be targeted. And then somatic mutation frequencies. I think everyone's familiar with the classic sort of mutation frequency graph from Gaddy Getz and others, where you see lung and head and neck cancer and melanoma with the higher mutation frequencies. This is from Bert Vogelstein's more recent report, which includes that same information plus colorectal cancer with microsatellite instability. And we know this is important with immunotherapy. I think it's well known now that, at least with the immune checkpoint inhibitors, the higher mutation frequencies or burden tend to correlate with response to checkpoint inhibitors. So if you look at microsatellite instability in immune oncology, this is from the TCGA, that you do have a subgroup of patients that have microsatellite instability, and as is well known now, in the paper last year, that those patients seem to do better. Microsatellite instability, whether it's Lynch or not, do better with a PD-1 inhibitor in terms of survival than those that are mismatch repair proficient. And then you say, well, this is really rare, and who cares about this? It turns out to be pretty common. This is a review by Dudley earlier this year, that these are the tumors that have greater than 10% MSI frequency. So colon, of course, is a big one, and endometrial, but there are a bunch of these. And these are cancers that have 2 to 10%, including head and neck cancer. So microsatellite instability is not uncommon. So I think the interest in immune approaches to this disease or to this situation are important. You've seen this one before. Again, I've talked about the germline, layering on the germline, the somatic, the interaction of the germline, somatic, and then the microenvironment. And I just want to say that when you're thinking about immunoprevention, again, we're talking about vaccines, there's a lot of interest in the immunosurveillance approach. And the sense is that if you can show evidence of immune surveillance, innate immune surveillance, and you can strengthen that with the vaccine, then you can enhance elimination. And one of the best, if not the best example that I know of is work here, really seminal work by Madav in myeloma, showing prospectively that patients who have anti-SOX2 T cells present before any treatment, just terms of following them, have a much lower rate of developing multiple myeloma than those that don't have this immune surveillance. You don't have the SOX2 T cells. I know he's making a vaccine to SOX2, which is a stem cell antigen, at least in this case, in myeloma. So I'll just make a few comments about Lynch syndrome, because I think it is a model of immune prevention that is fairly broad. Just to orient you, it is the most common hereditary gastrointestinal syndrome. 3% of all colorectal cancers, 2% of all endometrial cancers. I'm not going to get into this, but next-gen sequencing is changing everything. So it turns out that people in this field now studying familial syndromes are in this quandary where when you actually do next-gen sequencing, it's broadening the definition of these. So you can either characterize these syndromes based on actually what cancers families get, or you can characterize it based on the specific mutations that cause them. And I don't think anyone really knows the best way, but this syndrome is becoming much more common when it's done, when identified via unbiased next-gen sequencing. It's defined by a mutation in one of the DNA mismatch repair genes, mostly MLH1 or MSH2. At least in colon cancer, this shows you, if you look at the proportion of MSI tumors, this is all colon cancer, microsatellite stable. MSI is about 15% of colon cancers, and then you can see here a smaller percentage of those are hereditary or Lynch. So why is it a good model for immune prevention? Number one, you have a high-risk group. You need a high-risk group for any kind of prevention study because of the potential of adverse effects. Number two, as I'll show you, it's a good model of immune surveillance and escape. I'm not going to show you this data, but there's some really interesting data on the evolution of beta-2 microglobulin mutations being associated with infiltrate of T regulatory cells and so on, which is, I think, an important aspect of this, and of immune escape in general, microsatellite instability, and also as a model for high mutational, high neoantigen burden. Just a little hard to read this, but I think the point I want to make here is that it turns out that there's a specific type of mutations that occur, these frameshift mutations, in Lynch syndrome, and they generate these frameshift peptides, and these are the ones that are the most common. And it's easier to show here. The point is that you see evidence of immune surveillance in completely healthy Lynch syndrome patients. In other words, family members of someone with Lynch syndrome. It's remarkable. And so if you look at these healthy people, and these are just 3 different cases, but you can see here the amount of the frameshift peptide, specific T cells, and these here you see that even in no MSI cancer, but a Lynch syndrome. So this is really interesting, and so and there's speculation of why this is. There was a paper. I think people think that Lynch syndrome patients have all of these MMR deficient crypt foci, and so that they're generating these continual responses to, and they have these similar mutations, and that's what's causing these frameshift protein specific T cells. But the point is, you find them in healthy people at a rate that's not that different than people with MSH high colorectal cancer. And so you have this model where you're seeing these initiated MMR deficient clones starting really early, initiation, precancer, and evolving into cancer. And there are now some pretty good mouse models, like which is the one I guess this one. Where you're starting to see, you're able to model this. Some of the early models of Lynch syndrome were complicated and produced lymphomas and different diseases. So I think understanding this process is now becoming clearer. And then, so when I was asked earlier today, so okay, so high mutational burden, high neoantigen burden. This is all great for immune checkpoint inhibitors because they're kind of nonspecific, at least relative to vaccines. But how can you take that to a setting of prevention where you can't give an immune checkpoint inhibitor, I believe, at least not chronically? So what do you vaccinate against? Well, it turns out that there are a couple ways. In the MSI situation, it turns out that there's a characteristic pattern. Even though we think that's random, there's a characteristic pattern of frameshift peptide that exists. And so there's actually a vaccine being tested right now in patients with Lynch syndrome to 3 or 4 of these frameshift peptides. So again, a very different phenomenon to do it. Or the other way is to make personalized vaccines, which I don't think is that far off with the way as quickly as we do genomics. Now this is much more straightforward in some ways, more complicated in others. This is work from Liz Jaffe in a KRAS model of pancreatic cancer, and she's shown in the system that the first genetic driver is a mouse model, which in this case KRAS induces this inflammatory microenvironment. So the similar kind of microenvironment that we're wrestling with in cancer actually occurs very early in premalignancy, unfortunately. And so she's taken on KRAS, which of course no one's been able to solve in any setting, to my knowledge. But she made a listeria vaccine to KRAS and showed that if she gave it early enough, in early premalignancy, and she gave it with depletion of Tregs, with cyclophosphamide in this case, that you did see an improvement in this aggressive mouse model in terms of the development of pancreatic cancer. If you gave the same regimen in late premalignancy, it didn't work. Now, the challenge, of course, is that that how do you actually, somehow you have to be able to understand how the particular mutation relates to the microenvironment. And I just want to point out some work that a couple people that we recruited from the Broad Institute, Pablo Tamayo and Joe Mesroff, are doing. She's developing very sophisticated computational models to determine the actual state of a lesion or premalignancy, not just whether you're KRAS or not, but the state of it, integrating all this information and all the somatic information to develop more targeted approaches. And then if you want to make things even more complicated, right, so we have the microbiome, so that, which I consider as part of the tumor microenvironment. This whole area was actually reported initially by Michael Caron in a colon model. And obviously this is complicated. The microbiome can be helpful or harmful. And don't ask me how you can do this, but just like we talk about, computational biology, there's a way you can look at the TCGA, the already fixed specimens, and pull out the microbiome from there. So Del, you're nodding. Maybe you know how, but it's amazing what the technology can do. Because you think my issue would have to collect it prospectively and fresh to get the microbiota and the bacteria, but you can actually look at the tissue that's in the TCGA or other places and just pull out the different reads. And when you find something that's not human, then you see if it lines up and do it. So I think that we'll probably hear a lot more about this now. I don't think there's anything published on this yet, but it's an active area. So then I'll just finish with my favorite slide, which I still believe, and that is cancer prevention is the holy grail of cancer eradication. And all you have to do is take cervical cancer to realize that. And you can say, well, cervical cancer, but I believe that if we apply the same kind of next-gen sequencing technology, single-cell approaches, we'll be able to define premalignancy in very precise ways that we can develop vaccines too. But here you're talking about surgery, radiation, chemotherapy, and the most recent major report in New England Journal of Medicine, with or without Avastin, an advanced disease, showed a 2.5-month improvement in median survival. And in case you're interested, that wasn't significant. But if you look at prevention between screening and particularly HPV vaccines, there are 3 approved now. The vaccines have the potential to prevent over 90% of HPV-related cancer. And we just use cervical cancer as an example, but as Barbara and other people know, I mean, there's many other types of cancers, and a big one for the head and neck people is this oropharyngeal cancer. It's a huge problem. So, so if you look at this, I mean, I was going to make one of those seesaws to do it, but I don't think you have to think too hard to think that this is where we are in cancer by treating advanced disease, in this, at least in cervix, and here's where we get with prevention. So I think there's a lot of reason to stay with it. Bert Vogelstein had a great quote in his 2013 science paper saying, prevention's not going to be easy, but it doesn't mean we don't need to get there, because the payoff is huge here. And so I think that's my final slide. Happy to take any questions. Thank you.